Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 3. And we have been studying the prokaryotic cell, and we've been just studying the outside, the external structures in a prokaryotic cell, and we've gotten to the cell wall. Now, the structures we mentioned before, some bacteria have them, some don't, but all bacteria have a cell wall. So that's one of the defining characteristics of a prokaryotic bacteria is they have this cell wall. Before we start talking about cell walls, let's go back to cell membranes. Cell membranes is something you're familiar with eukaryotic cells. Both prokaryotes and eukaryotes have a cell membrane. That's the defining structure of a cell, and it just helps keep everything together, right? Phospholipid bilayer and cell membrane. So if we were to imagine that my balloon here, my visual aids, and my balloon here is a cell membrane, we know that as water goes into a cell, or air goes into a cell, that the membrane helps keep that water inside the cell, allows passage of water in and out, it's porous, that kind of thing. So this is a cell membrane. But you also know if I were to continue, I lost my balloon. If I were to continue to put air or water continued to rush into the cell is going to burst. It is going to lyse. It is going to break apart. Prokaryotic cells, who are out on their own in the environment without the protection of a body or a multicellular system in an organism, have a cell wall. Now, the cell wall is going to be a more rigid structure, and it's going to offer more protection from forces because it's rigid. Now, you'll notice, like my twine ball, it's also porous. Things still need to get in and out of the cell, so it allows that, but it gives a lot more framework and structure. This is going to allow the cell to take a certain shape. Unlike eukaryotic cells that don't have a cell wall and they can be all different shapes, a prokaryotic cell is going to have a specific shape. Some of them will be circular, and this is what we call coccus. Some of them will be rod-shaped, so little oval type shaped. We call that bacillus, but it's the cell wall that gives it its shape. And the cell wall is also important because when I take my cell and its membrane and water rushes in, unlike a eukaryotic cell that I can just continue to put water in and this will eventually lyse and burst, this cell wall will keep that cell membrane from lysing in a cell. So it's a lot of protection from osmotic forces from that. Okay, so let's look at our cell wall in a little more detail. We said its main function is to protect against osmotic forces. That's a eukaryotic cell membrane that's lysing there versus a prokaryotic cell that's protected. It also gives it characteristic shapes. We've mentioned the circles are coccus, the rod shaped is bacillus. Those shapes can be in multiple different arrangements. They can be in long lines, and we refer to this with the prefix strepto, so streptococcus for my circular shapes, streptobacillus for this, and then it can also be in clusters, and this is referred to as staphylo. So it gives us its characteristic shapes from that. The cell wall of a bacteria is made up of peptidoglycan. Peptidoglycan is something that we don't make. So we don't make it, we don't use it, which makes it actually a good target for drugs that are trying to get rid of the pathogen, the bacteria, without harming the host, such as penicillin. Penicillin actually works on a cell wall. So the cell wall can be a target of antibiotics and antimicrobial drugs. I mentioned it's composed of peptidoglycan. We've mentioned the two basic shapes. We can also classify cells based on their formation of their cell walls. And it was Christian Graham who came up with this procedure. It's a differential staining procedure, and it will allow us to sort these types of cell walls into gram-positive cell walls and gram-negative cell walls. And this is based on how they perform in a gram staining technique, which we'll learn more about in lab. Okay, so let's look at these characteristics. This is kind of a summary slide. We'll kind of come back to it in a little bit. Let's look at the characteristics of a cell wall. A gram-positive cell wall, it's going to have that cell membrane that's in yellow. All cells have a cell membrane, but a gram-positive is also going to have a very thick layer of that peptidoglycan. This is considered a gram-positive cell. The cell envelope, the part from cell membrane to cell wall, is in a gram-positive type arrangement, cell membrane and a very thick cell wall. Gram-negative is a little bit more complicated. It still has a cell membrane, all cells do. It has that peptidoglycan layer, that cell wall, but its envelope has one more structure. It has an outer membrane that's that we can see here in green. This is made of phospholipids and lipopolysaccharides. It is another layer of protection, and when cells have this, they are considered gram-negative cells. One other thing, because this is a membrane and because we have two membranes, the cell membrane and the outer membrane, we define the space between two membranes as a periplasmic space. So that's the name we give to the space in between these two membranes. So the whole structure is the cell envelope, membrane, wall, outer membrane. The space between the two membranes is the periplasmic space, and we have the cell membrane, cell wall, outer membrane. This is a gram-negative cell. Now, when you perform the gram stain, gram-negative cells will stain pink. When you perform the gram stain, the exact same procedure, gram-positive cells will stain purple from that. Okay, this is back to our picture of that we used for flagella, and I brought it back because you can kind of see this difference between a gram-positive and gram-negative cell. Here's my phospholipid bilayer, and then this is my peptidoglycan cell wall. Thin or thick? Do you have an outer membrane? So what kind of cell is this? Gram-positive. Gram-negative cell, we have our phospholipid bilayer. We have our cell wall made of repeated sugars. We'll look at that in a second. And then an outer membrane. So it is, see, it does have those phospholipids, and then it also has these lipopolysaccharides involved with that. So that's another view of the cell wall. What else? Let's do another view of the cell wall. Here is a cell phospholipid bilayer, cell membrane. I have this structure, gram-positive, gram-negative. Thick layer of peptidoglycan, so this is a gram-positive cell. Gram-positive cells also contain structures called teichoic acids and lipoteichoic acids. Teichoic acids are within the cell wall. Lipoteichoic acids extend all the way through the cell wall and into the cell membrane. So they're made up of these repeated phosphate groups, and that's their name. So gram-positive cells have this, gram-negative cells do not. Let's look at a gram-negative cell in a little more detail. We've mentioned cell membrane, cell wall, thin layer of peptidoglycan, and then this outer membrane. Oh, there's the periplasmic space between two membranes. This outer membrane. A gram-negative cell will also have these structures called porins that allow passage through this outer membrane. So these porins are through there. It's made up of proteins. And then it also has these lipopolysaccharides. This is a structure, a lot of different sugars, a lot of different things that make up with this that extend through all the cell gram-negative outer membranes. And I want to bring this to your attention because a portion of this lipopolysaccharide, this portion that's called lipid A, which is inside the outer membrane, is actually toxic to humans. Now, this isn't a huge problem when gram-negative cells come into our body. And remember, we have lots of bacteria in our body that are not pathogens and they're not damaging, and some of them are gram-negative. It's not a problem as long as the bacteria, the lipopolysaccharides, are connected and part of the bacterium. But say you have a gram-negative infection and you are going to treat that with antibiotics. What happens is these antibiotics, these chemicals, will kill these bacteria. The pieces will get degraded and chopped up, and then our body gets rid of them. But as it's getting chopped up, all of these lipopolysaccharides get chopped up, and a lot of this lipid A, just by itself, is released into our bloodstream and into our system. Now, this can be a big problem treating patients with gram-negative bacterial infections. Initially, you may prescribe an antibiotic. If you have a really severe infection, a lot of bacteria, prescribe a heavy-duty antibiotic, kills these gram-negative cells very quickly. A lot of this lipid A is released into the system. Now, the first 24 hours, your patient may respond, feel better, things are looking good, and then suddenly septicemia sets in, blood toxic shock sets in. You get this fever, all of these toxins go, and they have a negative effect, and you get a whole different set of signs and symptoms based on this lipid A toxin. This is when your patient takes a turn for the worst and suddenly succumbs to this bacterial infection from that. So again, knowing how these parts and pieces fit together influences how we treat them as well. All right, so that's part of lipid A, which is part of this lipopolysaccharides, which are found in the outer membrane of a gram-negative cell. All right, what else do we need to know about cell walls? All right, let's look at peptidoglycan a little closely. And all of these things we've visualizations, these diagrams, we've seen these repeated subunits, kind of looks like bricks from that. So let's look at that a little more closely. The peptidoglycan is made up of different types of things. It's made up of a polysaccharide portion. This is what the glycan comes from. And it's made of a protein portion. This is what the peptido part comes from. So it's made of these two repeated sugars or polysaccharides. First one is N-acetylglucosamine. The second one is N-acetylmuramic acid. They line up in alternating one from the other, and then they are held together by these peptide crosslinks, by these protein crosslinks. These proteins, these peptido crosslinks, these protein crosslinks only attach to the NAMs, to this one sugar, and that's how it goes together. So you can kind of see this. NAG and NAM repeat itself, and they make these long chains of sugars, and then the peptide, the protein portions, connect one NAM to another, front, back, side together, and basically you end up with, instead of long strings of sugars, which can easily be broken, you bind all these strings together into a very strong rope-like structure with these crosslinks, and this makes the difference, and this is why these cell walls can be so rigid and provide such good protection from prokaryotic cells. All right, now I always get the question during the semester, do I need to learn the full word? N-acetylglucosamine, N-acetylmuramic acid, NAG and NAM. I always tell students to learn the full word. It kind of helps you for that. If you're writing things down, I can give partial credit if you write N-acetylglutamic acid. If you can write it and you misspell, you add a couple of T's or something, I can give you some partial credit for that on exams. If you are trying to tell me NAG and you wrote NAP or something like that, I can't assume you know what you're talking about. So when you missed initials on things, I don't have a lot of grace on that because I don't think you know what you're talking about. If you knew the word was N-acetylglutamic acid, whether you can spell it or not, you're gonna get the abbreviation right. So challenge yourself, use the full word. Vocabulary makes you sound smarter when you use the correct vocabulary. So keep that in mind. Though NAG and NAM are pretty ubiquitous abbreviations, a lot of people know what that is. Okay, so now we have this structure, these sugar backbone and these peptide crosslinks. And so now let's go back to our summary slide. Gram-positive cells, we have that thick layer of peptidoglycan. They contain teichoic acids, both teichoic acid and lipoteichoic acid. They stain purple following a gram stain procedure. There are also some bacteria that have this structure with the thick cell wall, but within that cell wall, it also has a high amount of another substance called mycolic acid. Now, mycolic acids interfere with the gram stain procedure. We actually classify this. It is a gram-positive cell, but we can't stain it using a gram stain, and we actually have to use an acid-fast stain to be able to see these. And this helps these cells. Tuberculosis, that Mycobacterium tuberculosis, the Mycobacterium is part of this mycolic acid. Part of their reason that they're so well protected, so slow growing, is because of all this mycolic acid that's in there. So that's a gram-positive cell. Gram-negative, thin layer. There's a bilayer membrane. The outer membrane contains phospholipids and lipopolysaccharides. Part of that lipopolysaccharide is lipid A, which can cause its own set of signs and symptoms for a disease, and they will stain pink following a gram stain procedure. And remember, there is a periplasmic space between the outer membrane and the cell membrane of your cell envelope is defined as the periplasmic space. So, all right, I think that takes us through cell walls. This is a big deal, so make sure you understand that structure very well. It'll come back when we start staining and other types of things. Okay, let me know your questions. Talk to you later.